Hi guys. So I kind of threw a few extra things at you today, just because I want to keep us on par with the other CP Bio classes. So this is on your calendar for 2 days. So whenever you guys get the chance to take your notes on DNA replication, go for it. I'm just going to record me talking a little bit more in depth about DNA replication. You guys will be having an assessment coming up soon. It won't be like tomorrow, but I'm planning on Thursday of next week with a review day before we get into it. So just make sure you're really reading the book and reviewing the PowerPoint and going from there. So again, if you get to this today or Wednesday, just take your notes. I'm not going to check them, but make sure you're doing them. So we're going to start talking about DNA replication. We talked a little bit about the structure, how A and T pair together and C and G pair together. Those are our base pairing rules. And the discovery, really, we've spent a good amount of time on the discovery of the structure of DNA. So we're going to look at replication. Your test will kind of cover through replication, and then we will go into transcription and translation in your next chapter. So DNA replication, taking a closer look at it. The process of copying DNA is totally remarkable in its speed and the accuracy. More than a dozen enzymes work together with other proteins as well to participate in the process of DNA replication. When we get started, the replication begins at particular sites that are called the origins of replication. These are areas where two DNA strands are separated. They open up something called the replication bubble. In a eukaryotic chromosome, we can have hundreds, if not thousands, of origins of replication. But in prokaryotics, like a bacteria or an E. coli, they can only have generally one, sometimes two. Replication does proceed in both directions at the same time until the entire molecule of DNA is copied. So just visually looking, circular DNA from like an E. coli cell, hiccups, one direction, both directions, one bubble, all the way through until we have two daughter cells. Our DNA has several origins of replication until the entire molecule is replicated. At each end of the replication bubble is something called the replication fork. It's a Y-shaped region where DNA strands are elongated. Our first set of molecules that we're going to talk about is an enzyme called helicase. DNA helicase is responsible for untwisting the double helix, breaking those hydrogen bonds that hold the bases together. They are found at the replication forks because that's their job. They're untwisting and breaking the bonds of DNA. We also have single-stranded binding proteins that bind to and stabilize that single strand of DNA so it doesn't rewind together. And then we have I'd never say this one right. Topoisomerase corrects the overwinding ahead of the replication fork by breaking, swiveling, and then rejoining the DNA strands. It's going to make sure that DNA is not in a knot. Obviously, DNA gets wound very tightly into chromosomes. So we are unwinding, correcting the swivel, and then rejoining the DNA strand so it is one straight strand. Visually, that's his job out here. Holding it together, making sure it's just one wounded strand, it's not knotted. Our DNA helicase is here, unzipping in this direction. Our single-stranded binding proteins bind on both strands to make sure that they're not rebinding with each other. And then our primase is laying that RNA primer. DNA polymerase cannot initiate the synthesis of a polynucleotide. It can only add nucleotides to the 3' end of DNA. So this end here, okay? Notice there's no primer down here yet. We're going to talk about that in a second. The initial nucleotide strand that's created is a short segment of RNA called a primer. The enzyme called primase comes in to start an RNA chain from scratch and then adds the RNA nucleotides one at a time using the DNA parent as a template. The RNA primer is going to follow the same base pairing rules, A with T, C with G. The RNA primer is only about 5 to 10 nucleotides long and only starts at the 3' end to serve as the starting point for the new DNA strand. The enzyme called DNA polymerase catalyzes the elongation of DNA at the replication fork. Most of the DNA polymerases require a primer and a DNA template strand, which is the two things that we already have. The rate of elongation is about 500 nucleotides per second in bacteria, but only 50 nucleotides per second in human cells. But remember that human cells can have multiple points of replication starting at the same time. Each nucleotide that is added to the growing DNA strand is a nucleoside triphosphate, or DATP. It supplies the energy to DNA and is similar to ATP energy of metabolism. The difference is the growing sugars. DATP has deoxyribose, while ATP has a ribose sugar. Each monomer of DATP joins the DNA strand. It loses 2 of its phosphate groups, releasing energy in order to add that nucleotide. So here we see it come in. DNA polymerase grabs hold of it, attaches it, and drops out those 2 phosphates. DNA replication is called antiparallel elongation because DNA is an antiparallel structure, and it affects DNA replication. DNA polymerase can only add nucleotides to the free 3' end of a growing strand. Therefore, DNA strands can only elongate in the 5' to 3' direction. But one of our strands goes in the opposite direction. The strand that goes in the 3' to 5' direction is called the leading strand. DNA replication is able to occur continuously and smoothly towards the replication fork on this strand. So here's our leading strand. We get our primer. We're unzipping in this direction towards the left side of your screen. Here's our DNA polymerase 3 that starts adding on and is going to keep falling in this direction, as you see down here. The other strand is called the lagging strand. In order to elongate this strand, DNA polymerase must work in the direction away from the replication fork. The lagging strand gets synthesized in a series of small segments called Okazaki fragments. They're joined together by DNA ligase that acts as a glue to close the gaps between those fragments. So here, the first thing that happens is all the way down here. So the left side of your screen is where our helicase is unzipping. That's the direction of elongation. Here's our first primer that gets put down here. Primer gets put down, DNA polymerase 3 comes in and replicates. But now all of a sudden, we have to go backwards. Our primer has to jump all the way back here, put down a new primer, and lay down new DNA with DNA polymerase 3. We have another enzyme that'll come in and remove this primer, and then ligase can glue it shut. Again, looking at the two of them together, you have your leading strand up here, synthesizing in a very continuous way, and we have these gaps. Ligase comes in, we have DNA polymerase 1 that will remove that RNA primer, make a new gap. Ligase will jump back, seal this gap, while DNA polymerase jumps here, and it's just kind of chaotic, so it's lagging. It falls behind. The RNA or the DNA complex, it has a fancy name. The replication complex is called the DNA replication machine. The DNA replication machine may be stationary during the replication process, and the DNA may actually be moving through it. Recent studies have supported a model in which DNA polymerase and their nucleotides are being reeled in through the parental DNA and then extrudes or gets rid of the newly made DNA molecule. So this is what that would look like. Here's our complex, totally stationary. The DNA is being pulled through. This way you get one new strand out here and a second new strand out here. Another job of DNA polymerase is to proofread and repair DNA. DNA polymerase proofreads newly made DNA, replacing any incorrect nucleotides. Errors are usually kept to about 1 in every 1 billion nucleotides added, which doesn't sound like a big error, but it can be. In a mismatched pair of DNA, enzymes come in, correct the error during base pairing, and then seal it back up. DNA can be damaged due to exposure to harmful chemicals or physical agents such as cigarette smoke or X-rays. And it can also undergo random spontaneous change. Nucleotide repair uses an enzyme called nuclease that cuts out and replaces the damaged parts of the DNA. So here we have a damaged piece of DNA. Nuclease comes in, gets rid of it. DNA polymerase comes in, replaces the right ones, and then ligase comes in and seals the gap. There is evolutionary significance to altered DNA nucleotides. The error rate is very low, but it's not zero. The sequence of changes that may become permanent is actually how we got the blonde-haired gene. It was a mutation that ended up being passed from parent to offspring. These changes that are called mutations are the source of genetic variation upon which natural selection operates. And this is going to be the last slide that we talk about for today on this. Obviously, I want you to keep going in your notes, but I'm going to stop talking here. Replication of the ends of the DNA molecule. There's a lot of limitations of what DNA polymerase can do on a linear DNA molecule, like those of eukaryotic chromosomes. The usual replication machinery provides no way to totally complete the 5' end of DNA. So repeated rounds of replication produce a shorter DNA molecule every single time. This is not the problem for prokaryotes because they have circular chromosomes, so there's no end. But for eukaryotes, we lose a little bit of our DNA every single time we replicate. So just visually, that end, we lose it a little bit every time. Okay. Let me stop here. If you guys do have any questions, just send me an email, let me know. Again, watch this when you can. I tried to keep it short for you guys, but just let me know if you guys have questions.